just keep that in. So yes. So when it comes to genetic disorders, you get to see that traditionally there are 2 or 3 different types. Different types of genetic disorders based on their modes of inheritance also. So whatever is manifested and after a chromosome alteration is seen is known as a genetic disorder. So human genetics is the branch of genetics that deals with diagnosis, treatment and management of the individuals with genetic disorders. So this particular chapter is completely based on that only. In fact, all types of genetic disorders that are present. So this 46 chromosomes have around 10 to the power of 5 genes. That is nearly around 1 lakh genes that determine our embryonic development, the physical traits that we are seeing, and to some extent, based on this, even our behavior. So identification of the gene or the mutation that is taking place is very much necessary when identifying a genetic disorder, and it is quite complex. So when the human genome project had actually started, and then they completed it also, this has helped it, easier for us to identify the disease, prevent, and even certain times even manage the genetic disorders. Because once there is a genetic condition, either there is a fetal, for that matter, when it is expressed in during the fetal development time itself, there is either a miscarriage or term. The fetus does not come to term. And even with the genetic disorder, the child is born, there is a chance of the child not being able to become completely cured. They will have to, whoever has a genetic disease or a disorder will have to live and die with that particular genetic disorder without any kind of cure. So identification of the genetic disease or the disorder before the birth is known as parental diagnosis. And so this has helped in either, the continuation of the pregnancy or termination of the pregnancy. And if it is hereditary in nature, that is by passing from the parents to progeny, then the genetic abnormality is passed from the parents to the children basically. Or it can occur as a completely new because of some kind of mutation or exposure to radiation causes genetic alteration or deletion or translocation, because of which a disorder might occur. So when we come to this, we are going to see that we have a few terms that we have to know about, terminologies. So when you see hereditary means it is derived from the parents. Familial means it is transmitted in the gametes through the generations altogether. Congenital means present at the birth. So it can be congenital syphilis or toxoplasmosis. It is not exactly genetically determined. It is not something which is passed from the parents to the progeny, but it is possible for that particular syphilis to occur at the time of birth. So congenital means at the time of birth. Example, if you see, not all the genetic diseases are congenital in nature. That is, Huntington's disease is expressed or determined maybe around the 3rd to 4th. That is, when a person is in their 30s or the 40s of age, that is when it is expressed. So the genes are expressed at that particular point of time. So it is not exactly congenital. So they have given a few more terminology as given in your textbook. A dominant trait is expressed even if there is a single copy. You know that 2 copies of genes or genes are present. One is coming from the parent, mother. One is coming from the father, that is the autosomal genes, and the allosome genes are the X and Y in the case of male. X and X coming from, one X is from the mother, one X is from the father. So a dominant trait is one trait in which the gene mutation, even if it is present in one copy of that gene, is expressed. And a recessive trait is, so it is known as a gain of function basically. A recessive trait is something wherein both the genes, both the copies from both the parents are in the mutated or in the recessive condition. So it produces a non-functional protein. So if a cell contains at least one gene that codes for normal protein production, then the presence of a recessive gene will be masked. So in case that gene is encoding for a protein production, even with the help of one gene, because the other gene is recessive, then protein production is still taking place. So the other gene recessive is masked. But if at all that gene is not exactly, dominant in both the, copies from both the parents. So it is recessive copy. For example, it's a protein that produces hair. So if that protein is not present in both from the mother's copy and the father's copy, in the case of autosomes, then production of the protein that produces hair is not at all taking place, because of which no hair will be seen. It is just an example. Please don't quote it in the exams, because I'm just telling you so that you will be able to understand it in a more easier manner. So this is known as loss of function, because if there are two recessive gene cells, they'll not be able to produce the functional proteins. So the recessive trait is expressed. Now there is something called as a pedigree also. In pedigree, what happens is it is a diagrammatic record of the blood relatives of that particular individual and his ancestors. Therefore, a pedigree is constructed by collecting the information about all the person related by descent to the individual in question. So, for example, a child with Down syndrome has been born. So they will see, they are going to put in a chart basically, the child's parents and the child's siblings, the parents from the maternal side of the parents, the paternal side of the parents, how many brothers or sisters the child's father has, how many brothers and sisters the child's mother has, and then they go to the above generation, and this is how a pedigree chart is formed. So this is one thing. Now, when it comes to the classification of genetic disorders, you can see that you have either single gene disorders, or it can be chromosomal disorders, or it can be multifactorial and polygenic disorders. So three types are there. Again, this classification is based on whether the genes or the disease or the mutation or the disorder is present on the autosomes, that is chromosomes from 1 to 22, or the allosomes, that is the X and the Y chromosomes. So, and also the second factor that determines whether the classification is whether that gene is dominant or recessive in the expression. So again, in single gene disorders, you have, that is your Mendelian disorders, it is also known as. So you will have autosomal dominant inheritance, autosomal recessive inheritance. Then you have X-linked dominant inheritance, X-linked recessive inheritance. And then only Y-linked inheritance. You cannot talk about dominant and recessive because there is only one Y that is present, and that is being expressed in the males exclusively. Now, when it comes to chromosomal aberrations or disorders, this shows the chromosomal inheritance. And then you have disorders with multifactorial inheritance, that is the multifactorial and polygenic inheritance. So we'll just have a look at the multifactorial polygenetic inheritance right now. So if you see these influence, it is most common is that it is of a multifactor. There are many factors influencing this. They are because of the combined effect of many genes. So environment also plays an important role in this. So this the mutations, rather than being caused by one single gene, disorders such as diabetes, hypertension, cardiovascular diseases, multiple sclerosis is because of interaction of many genes that are present and many factors in the environment. So such polygenic disorders do not have any simple pattern as such, but they affect more people in some families than in some other families. Identifying the genes that are there that are responsible for this kind of susceptibility to this polygenic disorders has become better because of genetic mapping. And major progress has been made, and especially in type 1 diabetes. So examples again they have given regarding the multifactorial disorders. In your textbook at least they have given about cleft lip, neural tube defects, and pyloric stenosis also. Again, apart from that, you have diabetes, hypertension, gout, schizophrenia, all of these that are also seen. So individuals having a first degree or a second degree relative, that is first degree relative means the parents. Second degree relative means the grandparents or the cousins. So the degrees of relation is what do you call it? Means how well you are related to that particular person. So this increases the risk of having that particular disorder as such. So individuals having first or second degree relatives have have that risk of having that particular disorder. So risk again to the siblings or to the offspring is around 3 to 5%. And this risk increases when more family members are affected or the parents are also related. And multifactorial conditions relay, they run in the families with lesser risks than the Mendelian disorders, that is the single gene disorders. So pattern of inheritance is not exactly that predictable. And many multifactorial or polygenic disorders. So what happens is parents having an affected child, that is they have around 3 to 5% in the future pregnancies to have the same kind of affliction to the child as well. So you can use a mathematical model to predict if at all the next generation or the next sibling might have any kind of multifactorial or polygenic disorder. Again, sporadic phases, cases can also be seen, wherein, there is no previous history in the family, but a member is affected. So it is not safe to assume that the disease is not exactly genetic. Somewhere it can be there, or it can be caused by de novo, that is new chromosome abnormalities, new dominant mutation, or autosomal recessive genes, or uniparental disomy, germline mosaicism, or exposure to a teratogenic agent. So a teratogenic agent is something that causes, something like EDTA for that matter. And then you have your stains, some of them, they all are teratogens in fact. So they cause cancer or they cause mutations. They are mutagens. So in such cases, further investigation for medical and developmental history has to be seen. And then, as to it has to be seen whether it can be, whether the person in question can cope up with this particular disease or not. So this is about the multifactorial inheritance. They have given examples regarding gout, primary, secondary, morphology of arthritis. So I'm not going to go into these details. So right now, let us just get into the monogenic Mendelian disorders. That is the single gene disorders or the mutation of only one gene. So this is a Mendelian type of inheritance. And again, this change can be very small, that is altered chemical level at the level of a single gene. So the testing of such disorders is called as molecular genetics or DNA testing. This again can be categorized as 5, like I told you: autosomal dominant recessive, then allosomal, that is X-linked dominant recessive, and then you have Y-linked inheritance. So when it comes to this autosomal dominant inheritance, you can see that the affected individuals have one copy of the dominant gene. So, and the other it doesn't matter whether they are dominant or recessive, because it is inherited from one patient, one parent. So it can be heterozygous in nature, irrespective of whether the other gene has, that particular dominant or recessive gene. So both the males and the females, that is, both the progeny is affected. It is transmitted from one generation to the other. And 50% of the children are affected. So provided that the partner is not affected, because there is a 50% of chance for that particular gene to be coming into the progeny after crossing over. You can see that in your textbook, page number 99. So the affected father has one X chromosome and Y chromosome, that is, autosomal dominant, right? So it can be just basically alleles. So alleles are given. So the two alleles will be from the father and normal mother, two alleles. So 50% affected, 50% normal alleles are there in the father. So this, if you see the pedigree analysis, you can see that there is a 50% of that autosomal dominant allele to come into the child. So progeny. So because of which, if it is expressed, if it is present, it is like, the child or the progeny is affected. So some autosomal dominant disorders exhibit reduced penetrance or appear to skip generations because it is not expressed in some individuals who inherit the gene. Therefore, you have to draw this pedigree analysis chart to see whether that particular gene is prevalent. And it cannot be ruled out because only one particular family member is affected. So you can see, it is there in the family as such. And then find out the pattern. So example of this autosomal dominant gene is basically achondroplasia. That is one which is like short-limbed dwarfism, and Huntington's disease. And then it is expressed after the progeny becomes an adult, and it is a progressive neurological deterioration, and retinoblastoma, which is cancer of the retina. You also have familial hypercholesterolemia, that is one, Marfan syndrome. And so they have given the examples and what exactly happens during that. So all this is not exactly necessary. There are other cystic fibrosis also. So all of these are the autosomal dominant disorders. When it comes to autosomal recessive disorders, what happens is it forms the majority of the Mendelian disorders. Only the homozygotes are affected. So the parents are carriers. So if at all there is a carrier parent having 50% of that gene, then it is an autosomal. So you have around, if the parent is carrier, one allele is the gene that is affected, one allele is the carrier. Similarly with the parent, the mother also, that gives rise to carrier, 50% of carrier children, and one, that is 25%, or one child or one progeny is affected, and one completely normal child or progeny is seen. So only 25% of the descendants are affected. You can see this in your page number 101, in fact, in the pedigree analysis. Autosomal recessive trait, you can see that only 25% of the progeny is affected because of this. So again, examples are for this is phenylketonuria, cystic fibrosis, and thalassemia. So again, phenylketonuria is a deficiency of the enzyme phenylalanine hydroxylase, that is there. Thalassemia, wherein you have ability to produce the hemoglobin is decreased, because the hemoglobin shape instead of round becomes sickle cell, that is sickle cell anemia. Sorry, but thalassemia is the alpha and the beta chains in the hemoglobin, they are affected. So characteristics of this autosomal recessive pedigree, you see that it is an autosomal recessive disorder is one, expressed only when both the recessive pairs are seen in one individual. So only 25%. So it is a horizontal pattern of inheritance. So either gender can be affected because it is an autosomal trait, and both parents are carriers. Then you have a 25% chance in the pregnancy that the child can be affected. Then the frequency is higher in the case of consanguineous marriage, that is marriage within relatives, than in the case of marriage between unrelated individuals. So, and this character is frequently seen in both the males and the females. So example, like I said, thalassemia, sickle cell anemia, phenylketonuria, homocystinuria, cystic fibrosis, all of these are the autosomal recessive genes. And again, have just gone into detail regarding that. So absence of the enzyme phenylalanine pyroxenase and all this. Galactosemia is also an autosomal recessive disorder. Glycogen storage diseases, they include quite a number of type 1s, in fact. Von Gerk, Pompe's disease, then McArdle syndrome, all of these, deficiency of one of the enzymes that is seen. We'll come to this a little while later. Now let us see the X-linked diseases. In X-linked diseases, what happens is the X chromosome is involved. So most diseases follow an X-linked recessive pattern. So from it is transmitted by heterozygous mother to sons. So daughters can be 50% carriers, 50% healthy. Sons, 50% diseased, 50% healthy. Because the son is going to get the 50% is going to get the X from the mother and Y from the father. So if at all there is a son, if at all, the mother is a heterozygous carrier mother, and if that carrier mother with an X recessive chromosome comes to the son, then the son will become deceased. So there is a 50% chance for the son to be deceased. And daughters, what will happen is there will be two X chromosomes, because of which one from the mother and one from the father. So if the recessive X comes from the mother also and the normal X comes from the father, daughters will be 50% carriers, or they will be 50% healthy completely. And if this 50% carrier mother gives rise to a son, then that son will be 100%, he will be having that particular X-linked disease. So children of the deceased fathers, sons are all healthy because the father will not give the X chromosome. The father will only give the Y chromosome. All the daughters will be carriers. They are not going to be affected with the disease. But children of the deceased mother, sons will have the disease. Daughters will be the carriers. I hope you have understood this particular relation as such. And the X-linked dominant inheritance. Each individual has at least one affected parent that is dominant. Males are definitely affected. And when it is X-linked recessive inheritance, then there is no male-to-male transmission, but it passes on the males pass on the Y chromosomes to the sons. Females are not at all or very rarely affected. So the unaffected males they are not able to transmit this disorder as they do not have the abnormal X-linked gene at all. So carriers, again, like I told you, 50% chance with each pregnancy, and sons will inherit the gene, will be affected, daughters will be carriers. So female carriers might show mild symptoms as such, and because of the inactivation of one particular X gene, and a female carrier having this abnormal gene will be able to pass on the gene. 50% chance that the son is affected, 50% of the chance that the daughters are affected. So examples of this are color blindness, hemophilia A, and Duchenne muscular dystrophy, and hemophilia B, and Hunter syndrome. So hemophilia is a bleeding disorder. Blood does not clot because of the factor 8, which is absent or defected. Excuse me. Hemophilia B, there is a factor X. So you have blood clotting factors from 1 to 13, in fact. Hageman's factor, Christmas factor, factor 8, factor 9, then Hageman's factor. So all of these are involved in the clotting of the blood. Calcium also plays a role in this. So if at all in the case of hemophilia A, you get to see that factor 8 has a defect. Duchenne muscular dystrophy is a progressive muscular disorder. Basically, it is genetic, as you can already see. What happens is slowly the muscle, functioning will be atrophied, or the muscle gets atrophied. So again, you have to see that, in case of this, new mutations can occur, because of which this kind of disease can occur. It can also be a result of germline mosaicism, in which some of the mother's eggs carry this kind of mutation present. Rarely X-linked conditions are expressed in both the males and the females. In such cases, inheritance pattern can be drawn to see whether there is any kind of autosomal dominant single gene disorders are there, but there is no male to male transmission as such. It is always from female to male. So skip generation, as you can call it. Y-linked inheritance. Let us just have a look at it. Again, so please give me 1 minute, please. I don't think I have Y-linked inheritance over here. So you can just have a listen to it. Again, it is very small. The Y chromosome, we have already seen the sizes of all the chromosomes over here, of all the chromosomes that are present. So this small portion of Y chromosomes codes for something which is known as the testis determinant factor. So in case there is a Y chromosome, you know that it is always going to be transmitted from male to male. So character only seen in male exclusively. None of the females are involved or affected. So the father of each male also exhibits that character. So all the son of that particular affected male will have the same character. So example of Y-linked inheritance is hairy pinna. That is hair growing from the ears. So again, you can draw human pedigree, find out and all that. So this is actually limited to a number of individuals only. You cannot exactly draw and find out the basis of the ancestors right from centuries altogether. So the frequency of this recessive characters is basically seen quite a bit, and families are normally investigated only if there are affected children in it. So such families where both the parents are carriers, but fortunately none of the children are affected, are systematically missed. You will never know, because it is always a chance that is working out over here. Therefore, it is important for us to recognize the mode of the inheritance, so that, through the pedigree you can do it. And then, you can establish the fact as to why a particular person is infected. Now, when it comes to chromosomal disorders, you can see that chromosomal inheritance plays quite a bit of role, because of there are in any of these 46 chromosomes for that matter, you might be having some kind of chromosomal aberrations or disorders. So of which this 46 chromosomes, 22 pairs are autosomes, 1 pair is allosomes, you know that. Again, so 50% of the spontaneous abortions, 10% of birth defects, 25% of sexual differentiation, 20% of mental retardation, 65% of development in the females, reproductive defects are all because of chromosomal defects. So you have common examples that have been given over here: Down syndrome, that is trisomy 21, Edwards syndrome, that is trisomy 18, Patau syndrome, that is the trisomy 13. All of this have some kind of physical malformations, severe mental retardations, and some of them, they have been given, within the first, if at all the fetus comes to term, then you get to see that fetus will not survive beyond the first year of its life. And otherwise also there is going to be a severe debility for that particular individual. So common example of monosomy, if you see, it is like, Turner syndrome, which is 45X. So you have a missing X chromosome over here, resulting in short stature, webbed feet, reproductive organs are defected in nature, secondary sexual characters is missing, and X because that will be a female, Turner female. And you're going to get so the absence of the X chromosome, you will be having infertility, amenorrhea, that is lack of menstrual cycle, in fact, infantile genitalia, all of these, are seen basically. And it is a female because of the presence of an X chromosome. So structural abnormalities, all of these are also seen. Now, we have already seen this multifactorial and polygenic disorders. Now, we'll just have a look at the what do you call it, biochemical disorders, that is inborn errors of metabolism. So in this, what happens is every metabolism is related to a pathway. So in case one enzyme is deficient somewhere, there is you again have alternatives to that pathway, wherein even if there is a defect in one enzyme, it can be coped up. But because of an error, you can see that it is usually recessive in inheritance, so that both recessive parents, if they are seen, then that is when, you know, it is actually manifested. So if undiagnosed, it causes irreversible mental retardation and even fatality, along with neurological damage, physical disability. So early detective, detection of this particular, biochemical disorders is very much necessary. So, so you have neonatal screening programs for that right now, and you also have so all such kind of diseases or disorders like congenital hypothyroidism, all of it is known as congenital because by birth, and you can actually see that it manifests itself quite early itself, so that you will be able to identify that there is something different in nature. So congenital hypothyroidism, congenital adrenal hyperplasia, galactosemia, phenylketonuria, mucopolysaccharidosis, Gaucher's disease, amino aciduриas, and then you have, like I said, glycogen related or glucose related diseases. All of this can be known as a biochemical or inborn errors of metabolism. So you can see in this metabolic pathway, Beadle and Tatum had introduced the concept of one gene, one enzyme. So this has helped in identification of many of the inborn errors of metabolism. Of course, later you have a Beadle and another scientist giving one gene, many enzymes and other alternative concepts basically. But this gives the or helps us to find out the inborn errors of metabolism. So if A gives rise to B, gives rise to C, gives rise to D and E, and C gives rise to F and G. So what happens is, in creating product E from precursor A, you have intermediates B, C, D involved. So at each step, you have enzymes 1, 2, 3, 4 that is present. So if at all there is an enzyme number 3, which is an abnormal gene, then it gives rise to an abnormal D or lack of D altogether, product D and E altogether. And then what happens? Intermediate products, that is C, B, and A, they all are going to be formed. And then formation or the accumulation of abnormal metabolites, that is F and G, also takes place, which is not necessary in the first place, because I do not want products or metabolites F and G. I want product E, which is not coming in the first place, but I have extra metabolites that in the form of F and G accumulating. So these consequences are irreversible. So what happens is, this leads to other consequences such as neurological disabilities and mental retardation. So in case you identify that, yes, there is a product, there is an abnormal gene causing abnormal enzyme 3, then you're able to rectify that message and then either give enzyme 3 physically, that is intake, or at the same time, you're going to eliminate the F and G, which is also being produced. So this, if you can rectify the problem as such in the infancy itself, it is easier for the life to be a little more comfortable. So as you can see, your Archibald Garrard had published a book called as Inborn Errors of Metabolism. And over here, he had studied phenylalanine and phenylketonuria and alkaptonuria, 3 important diseases associated with absence of 3 important different enzymes. So let me see if at all I have phenylketonuria. Yes, please give me a So phenylketonuria is because of the deficiency of the enzyme phenylalanine hydroxylase that converts the phenylalanine to tyrosine. So the best way to maybe, rectify this at that particular stage is remove phenylalanine from the diet. So because of this, at least you will be able to see that, because there is no conversion of phenylalanine to tyrosine and accumulation of phenylalanine is seen. So if you remove or eliminate the phenylalanine from the diet, then what will happen is to some stage you can, prevent the, other effects as such. And then if more than one sibling in the family is affected and or if the parents are related, then that is consanguineous marriage. And in the recessive condition, this is going to be inherited as such. So they have given the following pathway also in your textbook basically. So inborn errors of metabolism, that is phenylketonuria, tyrosinosis, alkaptonuria, albinism, goitrous cretinism, all of these. So phenylketonuria is an autosomal recessive trait. So defective gene does not produce this enzyme phenylalanine hydroxylase, because of which conversion of phenyl pyruvic acid into hydroxy phenyl pyruvic acid is not taking place. So because of which what happens is phenyl pyruvic acid is getting accumulated in the blood. Severe mental retardation under 50 or under 65 is when you have the kind of IQ that is being seen because of this. In the case of tyrosinosis, you get to see that it is because of the conversion of beta hydroxy pyruvic acid into the 2,5-dihydroxy phenyl pyruvic acid formation. So this causes tyrosinosis. And if at all homogentisic acid is not converted into mallyl acetoacetic acid, then it causes alkaptonuria, because of which the urine of such kind of affected people turns black on exposure to air because of the presence of a substance which is alkapton, which is homogentisic acid. So, so this is passed on in the urine and later turns black on exposure. This is a recessive mutant allele H. In the homozygous recessive person only you will be able to see. So what happens is, even the ear, the cartilaginous region of the ear pinna also turned dark because of this. In the case of albinism, a person with a recessive genotype, that is AA, all of them small, then it is, this fails to produce an enzyme that is DOPA, that is 3,4-dihydroxyphenylalanine. This does not form or turn into melanin. So that kind of a person is known as an albino. Their skin is white in color. They are completely, the hair is white because there is no production of melanin. Melanin helps the skin to be a little like, the brown color, and, the melanin pigment gives the black color to the hair basically. And you can see that they're completely white or you can say fair, because it's not even fairness, because they lack pigments, they lack the melanin pigment. So, and they cannot be exposed to sun. They are very much susceptible to any kind of skin cancer, that they do not have any kind of UV protection as such, because of which more susceptibility towards skin cancer. All of this is seen in albinism. And they are hypersensitive in nature. They can get eczema and other kinds of skin diseases as well. So not very favorable for them, in fact. And when it comes to goitrous cretinism, such person does not have an enzyme for the conversion of tyrosine into thyroxine and triisothyronine hormone. So thyroid hormones deficiency is seen, because of which goitrous cretinism, that is dwarfism, is seen, and it is characterized by both physical retardation and mental retardation and hypertrophy. Thyroid gland wants to produce more amount of the thyroxine hormone and the triiodothyronine hormone, but it is not taking place, because of which hypertrophy is taking place and you get a goiter. All the neck area where the thyroid hormone is present becomes big. So this is all about the kind of disorders that are there basically. Now when it comes to teratogens, that it refers to any of the agents that are cause structural abnormalities because of which the fetus is exposed during the pregnancy. So a teratogen has been identified. For example, you have already learnt about it. You must have been exposed to this particular term, Minamata disease. In Japan, it was seen basically. So what happened was, there was a lot of mercury that had been released into the water bodies. So the fish got affected. Then, consumption of such kind of fish with a lot of mercury poisoning led to the, children being born with a lot of birth defects, especially floppy feet for that matter, and mental retardation. And all of this had taken place. So over here, the mercury is a teratogenic agent. So as you can see, ingestion of this contaminated fish during pregnancy was the primary cause for this condition. So similar results had been seen wherein ingestion of grain, which was with methyl mercury, which was used as a fungicide, was seen in Mexico and Iraq. Sudden appearances of the rare disorder can also raise suspicions of teratogenicity. So thalidomide is a human teratogen. It was used to treat the morning sickness. So this gave rise to, it was very good for the morning sickness. But then this thalidomide caused, after the first trimester, the children that were born had severe mental and physical defects. And some of them did not even come to term. So some of the teratogenic agents include infectious agents such as rubella, cytomegalovirus, varicella hive. Right now, I don't know how many of you know Zika virus is one. Toxoplasma, syphilis, all of these are the infectious agents which are teratogens. Physical agents include the ionizing agents and hyperthermia for that matter, and maternal health factors. So if at all, the mother has gestational diabetes or is it diabetic for that matter, maternal PKU, and environmental chemicals, that is organic mercury compounds, then bisphenyl or PCB compounds, then BPA compounds, herbicides, industrial solvents, all of these are teratogenic agents. And some of the drugs also cause problems. And you cannot exactly have alcohol also. Alcohol is also a kind of teratogen, especially the mother during the pregnancy, if at all, she partakes alcohol by mistake or whatever it is for that matter, then it causes issues in the fetal. So you can see that, fetal alcohol syndrome, fetal, hydantoin syndrome, fetal trimethadione syndrome, fetal warfarin. Warfarin is a blood thinner. Smoking. All of this have associated birth defects in the progeny. So public awareness has been of utmost importance in this kind of, situation. Now, when it comes to the parental diagnosis, you get to see that you have to do the genetic testing of the fetus. So again, congenital anomalies around 22-25% of the prenatal deaths, and the condition of the unborn fetus, all of this can be determined in this condition. So how are you going to do that? During gestation time, obviously, that is through the chorionic villi sampling or the amniocentesis or chordocentesis. That is this amniocentesis and chorionic villi sampling I have already discussed in detail in the first two classes. How it is performed, and with the I think the appropriate pictures are also there. I will try to send them again once I'm done sending. I think I should send you yesterday's notes as well as today's. If at all there is any kind of relevant PowerPoint, I will be sending it to you. If I feel there is no relevancy, please excuse me, I cannot exactly send you. If you want, you let me know whether you want this particular PowerPoint, I will send it to you. If you don't want, that's fine, because already you have quite a bit of information in your textbooks as such. So please let me know, put this in the chat box. If you want, I will definitely share. As such, it is available in the internet. Anyway, So chorionic villi sampling and amniocentesis have been discussed, and it is about, the removing of the chorionic villus using a biopsy, using ultrasound guidance. And then this procedure is performed around 10 weeks of gestation. Amniocentesis again at around 16 weeks of gestation. Chordocentesis only after the 17th week of gestation to remove the cord blood from that is, passing from the fetus to the mother. Again, when you're doing parental diagnosis, there is always a risk of miscarriage and a risk of infection to the fetus. So that has to be kept in mind. Now, when it comes to genetic counseling, it is an interactive session of the family with the medical genetics expert, so that analyzing, conveying the message about the genetic testing regarding the affected child, or the to be in case, what are the chances of the child to be affected, that is done by the genetic counselors. So they have they provide the information and then support the families who have members with birth defects or genetic disorders. Autism is also one you have, what do you call it? Yes, somebody has raised their hand. You can unmute and tell what you want to. All right, I will send this. Not an issue. Yes, Ms. Bharti, you had raised your hand. Is there anything that you want to share or tell us? All right, I'll continue with the lesson. In case you have anything, you can always message me. So this is about the genetic counselors. And when it comes to the preconception and prenatal risk assessment tools, over here you have to see that they have been developed so that, this increases the women's chance against having a very adverse pregnancy outcome. So you have to see the family history of the lady, or of the family that is involved in the pregnant women. The nutritional assessment has to be done in the case of phenylketonuria. If at all the mother is a carrier PKU, or in cases with women with PKU, they may not even know that they have PKU. So, however, that they'll be having special diet and all that later. You have such kind of information has to be shared with the doctors, so that this can they can at least prevent the what do you call it, passing of that particular disease to the progeny, or at least management of the progeny with the disease can be taken care of. So large quantities of vitamin A, if it has ingested, it increases the chance of giving birth to a child with malformation, similar to those seen with exposed to Accutane, which is a synthetic form of vitamin A. So if you keep on taking a lot of multiple, multivitamins also, that also causes an adverse effect on the child, in fact. Mega doses of vitamin D can cause severe infantile hypercalcemia. Elfin-like faces. Elfin means something like an elf, small face, which is triangular in shape. Pulmonic or supravalvular aortic stenosis, that is a cardiac defect basically in the aorta of the heart, and intrauterine growth retardation. So if it is a female, then you can get to see that there is, there is growth retardation in the formation of the uterus. When it is environment and occupational exposures, you get to see that, if at all you're exposed, you're working in a drug industry, you're working in a cotton industry for that matter, basic, or the asbestos industry, you're going to get something like the the black lung disease. If at all you're in a coal industry, and for example, bisnosis. So women who make stained glass windows, they're not they don't even know or don't realize that they're exposing their fetus to lead. So that is one risk. Or solvent fumes, or such kind of potential teratogens, cleaning fluids for that matter. Your bathroom cleaning fluid also has the sodium hypochlorite and sodium chloride in it. And this, finally, what happens is chlorine vapors are released. Even if you normally clean your bathroom also, you get to see that, you are ingesting those fumes, and you can feel that discomfort as soon as you ingest those fumes. So in case the woman is pregnant, then she is definitely exposing those fumes in the form of vapor, and this vapor gets into the bloodstream, gases exchange takes place, and through the chorionic villus, it gets into the fetus. So continued, maybe once does not make a difference. But if it is a continuous exposure, sometime or the other, the fetus is going to get exposed. So this teratogen creates a problem or a birth defect in the child without the parent or mother even knowing. So that has to be taken care of. So again, in the case of life cycle, when people hear this word genetics, they think about pregnant ladies, children with birth defects, childbearing couples. But you don't exactly think about the adults or normal adolescents for that matter, that is teenage children or adults for that matter. So whoever it is, end of the day, whatever be the situation, genetic counseling will always be helpful. That is what they're trying to tell with this particular paragraph, in fact. And what are the ethical and social implications of genetic disorders? We really don't know whether autism is genetic in nature, because I have seen children who are there are twins for that matter. One child turned out to be autistic, one child is completely normal. Otherwise, there were another set of twins wherein, both the twins turned out to be autistic in nature. Or the elder child is autistic, the younger child is completely normal, or vice versa, whatever it is. And more or less, it's seen in boys, and more of it than girls for that matter. So it is still being studied. We really do not know the kind of scenario for that matter. But all of this have a social implication. They have a social effect on the parents, on the children, and also how it is being dealt with. So genetic counseling then refers to giving of that information regarding a genetic or disorder or birth defect, implications for the current health, future development, or the psychological function, follow-up care of that particular individual. So again, in medical genetics, what happens is, what kind of, the diagnosis that has to be done, what kind of prognosis, what kind of treatment can be done, or how can that particular disease be managed. So this is seen in medical genetics. So, but a major difference is that diagnosis of a genetic disorder not only impacts the patient, but also the immediate and the extended family as such. So recognition of that particular, of the person with the particular genetic disorder leads to the realization that other family members are also at that particular risk itself. So that has to be kept into consideration, especially during genetic counseling and whenever you're dealing with a genetic disorder. Autonomy is something wherein, non-directive counseling is well accepted in the genetic community. Genetic counselor will be the information provider, and he is not the decision maker. The parents of the individual or the parents of the unborn child, of the fetus for that matter, when they're going for a genetic counseling, they are the decision makers. So information is presented in a very neutral manner, in a very non-judgmental manner. Then the patient or the family are made to make their choices. It can be very, it can be anything for that matter, but it is their individual choice. So by the same token, participation in screening counseling programs, that is carrier testing for Tay-Sachs disease, carrier testing for hemoglobinopathy, that is, disorders in the hemoglobin, parental MSAFP screening for NTD, all these different diseases, they all are voluntary in nature. All the pregnant women, for example, should be given the option of MSAFP. So you have to see for that. And then that option should be given, but it is up to the individual mother whether they want to go for that particular screening or not. The exception is that to this voluntary testing rule is the newborn screening, which is currently mandated in most of the states. See, when they talk about all this newborn screening, they're just going to do physical and the blood work and all of this is done basically. So that is one thing. And newborn screening seeks to identify disease condition, for example, phenylketonuria, sickle cell anemia, hypothyroidism. All of these is seen, the blood group is identified. And another important thing is the confidentiality or the privacy of the person or the parties involved. The genetic counselor should maintain that confidentiality. In majority of the cases, patient or the parents are more than willing to inform the at-risk family members about the diagnosis so that they can encourage them to take in the carrier or the pre-symptomatic testing. But sometimes what happens is, one person in a family has a problem with the with the child, or they have this, but they are not willing to tell that to that person's sibling so that they can go for carrier testing or pre-symptomatic testing. So if at all they don't do that, then this based on the fear that they're going to be stigmatized or they're going to be feeling bad, then this is one thing that is there because of any kind of family dysfunction. So primarily, the genetic counselor should tell that the patient's decision not to inform the other at-risk family members should be respected. However, rare situations where this confidentiality can be abridged is if at all there is any kind of voluntary disclosure that has been failed, high probability of any kind of serious harm that can be taking place for that particular at-risk relative. At such particular points of time, this confidentiality between the patient and the genetic counselor can be breached. Another area of concern, as they are talking about, is storage of the tissue samples. You have to write them properly and for family studies and all that. So tissue samples to be studied should be to determine the incidence of the specific disease in the population. If the tissue samples are used, then whether all the identifying information, you get to see, somebody, your relative tissue samples are there, then you will start, it is human nature to be inquisitive. But we do not want that. So just giving identification number instead of only all the information related to that. So they're thinking about whether to retain such information or to just, do away with them. Other researchers argue that restricted codes should be used so that people can be contacted if at all they are found to have or carry a specific gene, especially if any kind of disease prevention through intervention, that is disease prevention through any kind of medication or anything else for that matter is possible. Another issue of conflict that is being seen is between the individuals and their employers or insurance companies. Insurance company people are the first people to tell, no, this is not covered in the insurance. Sorry, we want you only to foot the bill. They're more than happy to, wash off their hands without paying us our insurance. We do not know all these clauses and the subclauses given in it. You already have this disease. Sorry, this is not covered in our insurance policy. So as it is done, and then we have to pay. For example, there are certain diseases which is not even covered by the insurance people. So people who choose to pursue this pre-symptomatic testing for conditions such as breast or ovarian cancer, Huntington's diseases, then they'll obtain information, and it can also be the interest to a third party. Then what happens? Your insurance is lost because of that. You go for this particular genetic testing. Then you get to know whether you are a carrier of that particular gene. So what happens is a third party is also involved, because of which they say that, sorry, you already have this gene which is positive, and you're positive for this Huntington's disease, so you're a carrier for it. So that is not covered, and you're in your insurance policy. So this kind of problem also can arise. So loss of this employment or insurability fear, is one of the reasons for people to actually forego or not go for genetic testing in the first place. So again, they have spoken about beneficents and the non-malfeasants. Simply put, what they say is attempt to do good, avoid harm. So genetic evaluation can be done instantly, like, then you'll come to know. Non-paternity can be uncovered when genetic diseases are, studies are done. So withholding information is also done with the intent of avoiding marital conflict and harm to the family unit. So example, whenever there is a genetic test done, and if at all, the paternity of the parent, is, what do you call it, in doubt. For example, in the case of, let us say, uh, there are people who go through, the IVF treatments and all that. Of course, sometimes what happens is, the father is known all the time. In case what happens is, sometimes when there's sperm donation that is being taken place. So then what happens is, it is a donor sperm that is coming in fertilized with the egg. So the paternity of the father is different over here. So this is because of which, either the acknowledgement for the non-paternity can be done or cannot be done. It is in respect with the female in question. Or in some other cases, the partner might be different, because of which mother can deny non-paternity. So further testing can be done to rule out uniparental disomy. So withholding such kind of information so as to avoid marital conflict, so that the family as such unit is preserved. That is one thing they can be done. Genetic counseling simply sees that it guarantees the provision of information which is sufficiently complete, reasonably accurate to allow parents to decide on a course of action. So child has a recessive genetic condition and father does not have this gene. Mother and father know that they have a low risk of recurrence. Mother is also told that recurrence risk with the biological father is around 25%. This scenario, withholding information does not actually negatively impact the parent's reproductive decision. So this is seen, and see, all of this mostly, is seen in the other countries. We do not exactly go ahead until and unless it is absolutely necessary for all such kind of tests. But then again, there is nothing wrong in going ahead with the genetic screening. Advent of pre-symptomatic screening, risk of real harm to the patient, that is, for example, depression, suicide, in this high risk, that is the HT patient for that matter, this is necessary. So that, stigmatization of the community can be reduced, or economic well-being is important in such kind of scenario. So all of these have to be discussed. Then the consent process should be taken, seen, and then the genetic program participation can be valued. In case of justice, so in genetics, what they mean is, it refers to the fairness, equal access to all the genetic services. So rural residents, that is people in the villages, they are quite at a disadvantage. Lack of awareness, basically. And new tests, as you know that, they're usually in developed cities and major cities, immediately available to the people outside is not exactly possible. So, and one more thing is the costs involved. These are quite expensive procedures. And the insurance systems are not exactly eager to cover genetic diseases tests, basically. In fact, I do not know how many of the insurance agencies are covering tests in the first place, because I have not seen so far. I'm not aware of actually, if at all insurance agencies are covering the tests, diagnostic tests costs. Usually they are the treatment cost is covered. So we do not know about the cost of the tests or the genetic disease tests. So conflicts may arise between the individual privacy, autonomy, and the state's right to know mandate policies for the good of the society in this case. So this is all about the genetic evaluation of counseling, which is done when appropriate. However, it is a consensus that it should be voluntary in nature, up to the patients, their families, to decide whether or not they can go for this particular genetic counseling. Role of the healthcare provider is that to gain the understanding of all the factors that are involved, so that whether the patient has to be known or it motivates or hinders the patient's participation in a genetic evaluation or a counseling program. This is used to help facilitate referrals. So this is all about justice. So again, like I said, to summarize this particular lesson, you can see that genetic disorders are classified as chromosomal, single gene, or biochemical disorders, and they occur as new events, and mostly they are inherited. Each chromosome has a lot of genes. For that matter, this 46 chromosomes have nearly 1 lakh genes being encoded on them. So there is always a chance for some kind of genetic disorder to take place. And changes in the gene may alter the production or the function of a protein, thereby manifests in the terms of genetic disorders causing physical or mental defects. Several modes are there in this genetic disorders. So changes in enzymes code for inborn errors, and accurate diagnosis, if it is done, will be able to help in improving the lifestyle. Then if you're working with a patient or a family with a possible chromosome abnormality or a single gene disorder and a history of 3 to 4 miscarriages is seen, and then how other family members are affected is also taken into evaluation or considered. And then individuals who have high risk ethnic groups. There are certain groups who are actually having sickle cell anemia, thalassemia. Within them, some of them, they're there. So such kind of ethnic groups, or those who are having a consanguineous relationship, that is those who are married within the families, the first cousins, or the having such kind of relationship is known as a consanguineous relationship. They also benefit from the genetic evaluation and counseling. So if at all you're dealing with a patient having an abnormality, that is when you cannot, that too when there is a genetic disease, you should collect additional information, so that in case you can determine if at all the genetic referral is appropriate. You have to take into consideration the teratogens that are present at home and at workplace, the kind of effect that is going to have on the fetus. The time of exposure also makes an important determinant. First half of the pregnancy, that is the first trimester, and subsequently the second trimester also, is quite important and very vulnerable. So it is essential to be extremely careful during this particular point of time. And then you have to go for diagnosis, management, then to family members, and then for family members you have to do counseling, then risk counseling, pre-symptomatic diagnosis, and in general basically you have to do carrier detection and newborn screening for all the general population. So this is all about your genetic disorders and how to do diagnosis, about genetic counseling as such. So in the next class, I will start with mutations and the repair of DNA. And so that's what. We have 15 minutes of time. Actually, I want to start off this lesson as well. Simple reason being, we have 10 classes and 16 topics. And so far we've had how many classes? First, 7 and 8, 3 classes. And I could complete 4 topics so far, up to genetic disorders. So basically developmental biology has a few topics which are slightly smaller in nature, which can be completed in 2 classes. So I will try to take in those. I will club 2 classes each and take 2 units each and take in 1 class. Right now, what I'm going to do is start with mutations and repair of DNA, because we have another 10 minutes of time and I don't want to waste. Give me a moment. So when it comes to mutations and the repair of DNA, You get to see that effects of mutation causes DNA damage and cell inherent, the capability of the repair. So DNA undergoes frequent changes, either chemical or some kind of a mutation takes place. So, and at the same time, those genes that are not exactly repaired, this leads to genetic mutation. So this mutation is a failure of the DNA repair. Since the development of a potential of an organism is determined by its genes, DNA must necessarily change, mutate as organisms evolve. So that is where your DNA repair genes also come into place. So again, they have just given the same thing. For example, that genes are made up of DNA, and DNA has the nucleotides basically. That is sugar plus phosphate group, and you have around sugar nucleotide having a base, 4 different bases: adenine, thymine, cytosine, and guanine, and they are referred to by the letters A, T, G, C, and G. And so DNA is made up of 4 different types of nucleotides having bases A, T, G, and C. So set of 3 nucleotides is known as trinucleotide, that is TAG, GCC. These are the examples that have been given in your book. So a gene has a set of nucleotides, 3 nucleotides in a particular sequence in a string. So they are made up of proteins again. So proteins, you know that they are made up of amino acids now. Now, amino acids, we have been studying from since our childhood. They are the building blocks of proteins. Arranged in a specific order, you have nonsensical proteins for the stop, which are the stop codons, as you know. So a trinucleotide is therefore known as a codon. Codon can be a word for an amino acid, and other codons contain this information for the cell to start making the protein or to stop the protein. So you have UAA, UAG, and UGA, which are the terminal codons, which are known as the nonsense codons, which is when when the protein encoding comes, when the ORF comes over there and stops, it senses that it is a stop codon, and then that is when the protein synthesis is stopped. So sequences of nucleotides in sets of 3 in a gene provide the code for the amino acid sequence. So this is known as the genetic code. So like they said, genes or codons is the recipe for a protein. So the code is translated by the cells so as to for the protein synthesis. So now when it comes to the importance of the genes are the strings of codons arranged in a specific order. So again, they have mentioned the same thing. Again, I'm not going to go into it. When it comes to the importance of reading this genetic code, first step is in determining the cause of a genetic condition to locate that particular gene involved. So once this particular gene is located, you have to see the coding sequence of that particular gene in that particular person's cell. So, and then you have to see whether it is in the correct order, that coding sequence, or whether it is mutated or different. So the ability to identify a particular faulty gene, define the exact type of mutation, can lead to the diagnosis of the genetic condition in that individual or in that baby. So what happens is, in other cases, this can be the results of the genetic test shows that that person is predisposed or is at a higher risk of developing that particular condition. So the effect of having a faulty gene on a person's growth, development, or health is determined by the type of mutation in the gene, where that particular gene is located on the chromosome, and the impact of if the gene is located and if the gene has a issue, what does it have? The impact of the gene product. It can be protein synthesis, it can be lipids, whatever for that matter. So the gene product's impact is more important. So that is the about the genetic mutation and its importance of reading the genetic code. So in the types of mutations that are present, Uh, so I'm just like kind of trying to see basically like the kind of mutations that are present, and in case if at all we are able to. So you have single base substitutions, which are actually known as the point mutations, and then you have nonsense mutations, you have silent mutations, splice site mutations. So these are the different types of mutations that are present. So what I'm going to do is I'm trying to check in if at all there is what do you call it? Right. Please give me one moment if it's possible. What I'm trying to do is download a PowerPoint basically so that you can get to see with the fact that we have another 5-10 minutes in the class, because we are short of time. Right. Resolution. So change in the nucleotide sequence of the genome of an organism, virus or extra chromosomal genetic element for that matter. Types of mutations are substitutions, insertions, deletions, frame shift mutations. So insertion deletions are known as indels, in fact, frame shift mutations. So let us just look at the substitutions. So substitution is single, it can be single based also, or it can be nonsense and silent mutations. We'll just have to have a look at it. So what happens is in this single base, say for example, A is there over here, adenosine or adenine. So what happens is this is being substituted by G when it is cut. So they are known as point mutations. So purine, that is the A or G, is being substituted with pyrimidine, or pyrimidine is replaced by the other substitution. This is known as a transition. Purine is replaced by pyrimidine, that is A is replaced by C or T, and then C or T over here, or vice versa. Then it is known as transversion. So if you have understood, purine is replaced. So this is a transition substitution example that is being given over here. Simple. If A is replaced by C or T, then it is known as a transversion. So what happens is over here, the protein is going to be different. It can be for a different altogether. You can see a textbook, it's got a better example and better understanding. So what happens is instead of histidine CAT, you're getting a proline. Replacement of the single nucleotide A with C has resulted in a different amino acid altogether. So instead of a histidine, you're getting proline. So this is the kind of substitution that is taking place. And in sickle cell disease, for example, replacement by A by T in the 17th nucleotide, gives rise to, instead of glutamic acid, it gives rise to valine. So in the 6th amino acid, you get valine instead of glutamic acid. So this results in sickle cell disease. In the case of nonsense mutations, with nonsense mutation, what happens is new nucleotide changes a codon specified for stop codon. So stop codon, that, TA, TAG or TGA for that matter, instead of CAG. What is happening is on a transcription translation. In this, the translation part, instead of you're getting CAG, you're getting it as TAG. So UA, UAG, UGA, TA, TG, TAG, TJ, all of these are stop codons. Please don't get confused. So in this, if at all a stop codon comes, instead of further protein synthesis, this synthesis is being stopped altogether, because of which you do not have a further protein only. So this kind of mutation is seen in cystic fibrosis. In case of silent mutations, I don't exactly have those over here. I just have a couple of um, mutations over here. I'm trying to see if at all there's anything else that has mutations and the kind of substitutions. Anyway, in case of silent mutations, you can see that most amino acids are encoded by different or several different codons. So third base in TCT codon for serine is changed with any other third base. Serine will still be encoded. You can still get the serine only. Such mutations are said to be silent in nature because they cause no change in the product and they cannot be detected until and unless you do an mRNA sequencing or the sequencing of the gene. In the case of splice site mutations, what happens is introns, which are the non-expressing part, are removed during the translation, and mRNA, which has only the expressing part of the gene, that is the exons, are expressed. So removal of the intron, which actually gets not expressed as pre-mRNA, is being processed to form an mRNA. So this has to be done with great precision, right? Introns have to be removed, exons have to be present. Coding part should be present. So if any kind of mutation alters this particular sequence or the signals basically, and intron is not getting removed, what happens is it will be there in the final RNA molecule also. So this alters the sequence of the protein that is coming out, which again is not necessary for us. Now, in the case of so this is all about splice site mutations. In case of insertion deletions, which is known as indels, what happens is extra base pairs are inserted into the nucleus in the DNA. Or insertion, see, that is what is happening. Instead of UAC, tyrosine, what is happening is UUA, you are having UUA over here again. So there's an insertion. So because of which what is happening is you are getting a leucine and arginine over here instead of only tyrosine and glycine. So this again causes issues. Or it can be that deletions will occur because of which, the sequences, do we have deletion over here? I'm trying to see if at all there is any deletion. You can see this is all, they have also mentioned about inversion. Inversion is when you have a DNA sequence is reversed. It can occur amongst a few bases within a gene or longer DNA sequences. So see here, C and G, the pyrimidines are being inversed with the, purines. So this is an inversion that is occurring. And deletions are mutations in which section of the DNA is lost. So when this is done, you have a stop codon, protein will be stopped altogether, the protein sequencing. So you're not going to get a malfunctioning protein as such. You also have duplications, wherein what happens is you also have in which there is a doubling of that particular genome taking place. So whenever gene duplication has occurred during this evolution of eukaryotes, genome analysis can be done so as to check about to this particular mutation that is taking place. So such gene duplication can be beneficial also, and this acquires a new function altogether. So this can be seen in the evolution, especially during the adaptive evolution, or it can offer redundancy and also have an evolution. It is evolutionarily significant in nature whenever a gene is getting duplicated. And you also have frame shift mutations. I will have to come into that. It is when the three codons, insertions and deletions, can alter a gene so that the message is no longer correctly read. So over here, I start with CAA-TACCT. What is happening is addition is taking place over here and a deletion is taking place over here, because of which the reading frame, you read 3 amino acids at a time or 3 nucleotides at a time to form the amino acid. So when you're reading this, this is known as the reading frame when during the protein, during the transcription and the translation. So this causes a shift in the frame, the reading frames, the sequence. This causes frame shift mutations. There's one more mutation as such, which is translocation. That is transfer of one piece of chromosome to a non-homologous chromosome. They're often reciprocal. We have already read that in the last class. So a gene will break, and this is going to occur within the gene. So that particular gene's function is destroyed over there, and this can come under the influence of different promoters and enhancers so that their expression is altered altogether. So this is seen in Burkitt's lymphoma, for example. And breakpoint can occur within a gene, so that what happens is a hybrid gene is formed. So this is seen in Philadelphia chromosomes, which is seen in leukemia, chronic myelogenous leukemia patients, because of the result of a translocation. This produces a compound gene, that is BER-ABL gene basically. So this is about the different kinds of mutations that are present. From the effect of the mutation on the gene product onwards, what I'll do is we will do this in the next class. All right. Now, thank you so much. We will meet in the next. We will meet next week. All right. Thank you.
Thank you, ma'am.
Thank you.
Thank you, sir.
Thank you, ma'am.
Yeah, sure. Thank you.
Thank you, ma'am.
Thank you, ma'am. Ma'am,
Ma'am, please send the PDF, ma'am.
ma'am, please send PDF. Hello, ma'am.